Hello, everyone. Um, Today I'm going to give you a quick overview of the microscope. The microscope is used obviously to look into very small structures that you cannot see with your naked eye. So it lets you see very small, like bacteria, virus, and whatnot, cells. This is a very important instrument for any science student, be it in anatomy, biology, microbiology, etc. It has two major functions. The two functions are magnification and resolution. You would see that in the very first page of your lab manual, the exercise 2. Magnification obviously means it would enlarge the object, and resolution means clarity. It not only would enlarge a tiny cell, but it would continue giving a clear appearance of those tiny structures. Imagine the cell, it has even a smaller structure called nucleus inside. When you enlarge, it could easily make it blurry, but the microscope has the ability to avoid blurriness, so you'll see clearly much bigger cell, nucleus, etc. The microscope can be simple or compound. The simple type, if you have seen in pictures or you have one or you have used one, would have only one lens. We won't go into that. We need this one. This is called a compound microscope because it has two sets of lenses. I hope you can see the two sets of lenses. One is the ocular lens. This is where you put your eyes. And the other one, or the objective lens, this is where you put the object, which is a slide. As you can see, the ocular lenses could have been adjusted. You have a question about the magnification power of these lenses. It's actually printed on the lens. The ocular lens can magnify 10 times. The objective lenses are screwed onto this revolving piece. What I'm touching and holding, that's called a nose piece. It has these three objective lenses. The shortest one is called the scanning objective lens. It has a red ribbon around. Scanning has 4x written on it. That means it can magnify 4 times. The next one is the yellow ribbon one. That's the low power lens, and it can magnify 10 times. And the longest of the three is the blue ribbon one that can magnify 40 times, and it's called the high power lens. So scanning, low power, and high power, these three lenses are the objective lenses screwed onto the nose piece. This is the stage. The stage has this metal clip device, has a slot where you would put the slide. To begin looking at a slide, you must always start with the scanning in place. And the slide goes in here and snugly fits in there. Here is a movable device. This one moves the slide holder side to side and back and forth. Underneath the stage, you have a condenser. The condenser aligns the light beam from the illuminator or the substage light. This throws light up, it comes through the condenser and then comes out through the slide to illuminate the object you're going to look at. And then in the condenser, you have this another movable piece. It's called the iris diaphragm. This thin, easily movable, black needle-like structure. This one controls how much light will come through the microscope. If you would have done it here in the lab, you would see how if you have the iris diaphragm open and allow too much light to come in, a white transparent object like a cell would actually be invisible. You wouldn't be able to see it until and unless you close the iris diaphragm completely and create a contrast so you could see the object. I already mentioned the substage light here, and then you have the two big and small knobs. The big knob is the coarse focus knob, only to be used with the scanning objective lens. And the smaller of the two, if you can see now, the smaller of the two, this is the fine focus knob. So the coarse focus knob you use with the scanning objective lens. Once you have focused the object, by moving this, as you can see, if you move the coarse focus knob towards you, it lowers the stage a little, just a little move, and you would see the object. And then you would fine focus it to make it clearer. And when you are ready, you would move the nose piece to get into the next one. A little bit of light adjustment, a little bit of fine focusing, and you would see the object even bigger. And then finally, you would have gone to the high power and then see the largest possible with this microscope. So you have to determine, like you're looking at the object with the high power, what would be the total magnification? To determine the total magnification, you would have to multiply the ocular lens magnification power with the magnification power of the objective lens. So when you have the high power lens in place, its own magnification is 40 times, but the total magnification would be 400 times. In other words, with this instrument, we can make an object 400 times larger, and that is enough to let us look at a cell membrane, nucleus, even chromosomes inside the nucleus. Obviously, you can see the power cord nicely wrapped in the bag that I didn't mention yet. Unwrap the cord a little, plug it right in here. Here's the power switch. And you can see, I hope you can see the light coming. This is the controller for the light. Go high and you can see the light coming, and the light goes to the condenser. You can see a lot of light coming through here now. And if I close the iris diaphragm, very minimal light would come here and create a contrast so we can see transparent objects. So you would start with the light a little dimmer. You would put the lens here, the slide here, adjust the slide, bring it down to the middle. Put your eye here on the lenses, not letting your eyelash touch the lens, right about an inch and a half away from the ocular lens. And inside you would see a pointer, a built-in needle-like structure that allows you to pinpoint a structure you would want to see, or you would want your instructor to look at and identify for you. And we use that pointer and put it on a specific structure, like a nucleus, and ask you to identify that structure. It is so unfortunate that you won't have the opportunity to use the microscope here, We do a overview again in Anatomy Physiology 2 classes. So hopefully in the spring we will come back to the campus and you will have this amazing experience of handling a microscope and looking at the very small structure. Throughout the lab manual, you will see a lot of these pictures of the slides that show you all these very small structures magnified, and you may see as a footnote under the picture, it says under 40x magnification, so you know which power was used to show the object. That's it. Hope it helps.